Hi, I'm David and this is the Biology Classroom. This is a paper discussion video, so get ready with this paper and let's go through the questions and answers together. Question 1 is about the cell cycle. A wants you to state the proteins complex with DNA. It is the histones. B asks you to complete the figure to produce a labeled diagram of the metaphase stage of mitosis in an animal cell with 2 chromosomes. The 2 chromosomes should be at the equator of the spindle or the metaphase plates. Each contains 2 sister chromatids as DNA replication occurs during the interphase. There will be 2 pairs of centrioles at the poles. Spindle fiber originates from the centrioles, connects to the centromere of the chromosomes. You have to label at least 3 structures to get the mark for labels. C. Outline the changes that occur to the structure and behavior of a chromosome from the start of the S phase to the end of the interphase and during the prophase of mitosis. The S phase is the synthesis phase. This is when DNA is synthesized by DNA replication. DNA replication is done semi-conservatively. The new DNA molecules associate with histone bodies, forming chromatins. Each chromosome will now have 2 chromatids. They are called sister chromatids. G2 phase occurs after the S phase, but the events that take place are not related to the structure and the behavior of the chromosomes. In prophase, chromosomes condense. They will become visible. If you view the cell under a microscope, you can see the sister chromatids. The centromere of the chromosomes will be attached to the spindle fibers. The attachment is possible due to the formation of kinetochore. A kinetochore is a protein complex that forms on the centromere. It allows microtubules to attach to the chromosome for proper segregation during cell division. Question 2 provides some information about keratin. The question mentions that the alpha and beta keratin contain many alpha helices and beta pleated sheets. These are the secondary structures of proteins. B suggests why it is possible for a protease to act on different types of protein. To hydrolyze protein, the active site of protease must bind to the amino acids where the targeted peptide bond is found. Many different types of proteins may have the same amino acids in the area where the active site binds. The protease will always hydrolyze peptide bonds between the same amino acids. Another possibility is that the active site has some degree of flexibility. It changes shape slightly for binding to similar but not identical substrates. The proteins that can be hydrolyzed by this protease may have similar shapes. So, all of them can fit the active sites even though they are not the same. The active site has a complementary shape with more than one type of protein. There is a mark for AVP. For example, some large enzymes or enzyme complexes may have more than one active site, so they can bind with different substrates. C suggests features of keratin structure that contribute to its stability. At the start of this question, it was mentioned that keratin is a structural protein. Most structural proteins are fibrous. So, you can use your knowledge about collagen to answer this question. But be careful that, even though they are both fibrous proteins, they are not identical. So, not every feature of collagen can be applied here. Unlike globular proteins, fibrous proteins do not have a hydrophobic core and hydrophilic amino acids facing outwards. So, they are insoluble and do not dissolve easily. Since keratin has a high proportion of cysteine, there will be a high proportion of disulfide bonds. They form between the sulfur in the R groups of cysteine. The presence of hydrogen and oxygen in a protein allows a high proportion of hydrogen bonds between polypeptides. When many hydrogen bonds form between them, the stability increases. Fibrous proteins usually also have a high proportion of hydrophobic interactions. In general, there are many amino acids with hydrophobic R groups. The polypeptides may be closely packed like how they are in collagen. Spaces between the molecules are minimized, contributing to a tougher physical structure. There is a mark for AVP. You can talk about the arrangements of the molecule, which makes the peptide bonds inaccessible. This makes it difficult to break the bonds. Besides, many amino acids in keratin may have small R groups. These small amino acids allow the polypeptides to be tightly coiled together. Figures 2.1 and 2.2 shows the effect of temperature and pH on three types of keratinases. D wants you to complete the table showing the working range of temperature and pH where at least 60% of the relative activity is obtained. Draw a line at 60% in both graphs and you can read the range easily. To know the enzymes with the widest working range of temperature and pH, you have to calculate the difference between the highest and lowest values of the range. E, with reference to figure 2.1 and 2.2, discuss the advantages and disadvantages of using K12 and A22 in the detergent industry. When answering this question, you must use the information provided in the figures. Do not suggest features which are not evident in the question. The first mark is given to an advantage related to the working range of temperature. Both enzymes work at a wide range of temperatures. Quote the temperature range to support this idea. You can also use the activity of the enzymes at high or low temperatures to discuss their suitability in hot or cold water washers. The second mark is for the disadvantage related to temperatures. State the temperature and activity where these enzymes have low activity to get this point. The third point is for the advantage obtained from the pH graph. The question says that protease in washing detergent should have high relative activity in alkaline conditions. So, you can use the information in the pH graph to discuss how both enzymes are active at an alkaline pH range. The fourth point is for the disadvantage related to pH at which they are active. Even though they are both active at a lower alkaline pH range, there is a decrease in activity when the pH continues to increase. You can use their activities at high pH values to show that both enzymes are not effective in highly alkaline conditions. The last mark is given for any wash feature linked to any point that you have mentioned before. For example, if you state in your first point that the enzymes work well at high temperatures, you can link this to the benefits of using hot water for washing. You can also compare the features of the enzymes such as thermal stability and pH stability. Question 3 is about a type of cell called hepatocyte found in the liver. A. The main artery that transports blood to the hepatic artery is the aorta. The largest artery carries oxygenated blood from the heart to the rest of the body. B shows a blood vessel called a sinusoid and a phagocyte called a Kupffer cell. 1. Suggests one advantage of having large gaps between the endothelial cells. The gaps in blood vessels allow a high rate of material exchange between the blood and the surrounding tissue, allowing substances to move more easily in and out of the blood. They do not have to cross a cell layer. In this case, they do not have to cross through the cell surface membrane of the endothelial cells, making a more rapid movement. Larger molecules, such as plasma proteins that are found in the blood, can pass through and reach the tissue. Kupffer cells can also pass through into the tissue fluid. They can perform phagocytosis in the tissue fluid. 2. Describe the mode of action of a Kupffer cell in removing and breaking down a damaged red blood cell. Phagocytes can remove damaged body cells by performing phagocytosis. It is a type of endocytosis. They extend their pseudopodia around the cells and engulf them. Pseudopodia are temporary extensions of a cell's cytoplasm. The rest of the points about the events afterwards will give you 3 marks maximum. That means you must have one of the first 2 points to get the full marks. Once the red blood cells is inside, the membrane of the Kupffer cell fuses to form a phagocytic vacuole. You can also call it a phagocytic vesicle or phagosome. Lysosomes will then fuse with the phagosome. They contain hydrolytic or digestive enzymes. The fusion allows these enzymes to be released into the phagosome. Digestion occurs inside the phagosome. For example, hem and globin are produced due to the breakdown of hemoglobin. Amino acids are produced due to the hydrolysis of proteins. There is a mark for AVP. You can talk about how the Kupffer cell recognizes the red blood cells to be engulfed. Figure 3.2 is an electromicrograph of a part of a hepatocyte. We have some information about the peroxisome. C1 wants you to describe the evidence visible in Figure 3.2 indicates some of the functions of a hepatocyte. You also have to add labels to identify the location of any cell structures that are part of your evidence. At the beginning of this question, 3 functions of hepatocytes are listed. The synthesis of triglycerides involves the smooth endoplasmic reticulum. It has a tubular appearance with no ribosomes. They are found at the top left corner of the photomicrograph. Naming and labeling the structure plus any one detail will give you 1 mark. The rough ER is responsible for plasma protein synthesis. It is an interconnected network of flattened sacs that has ribosomes on its surface. At the bottom right corner of the picture, you can see those flattened sacs with dark spots on their surfaces. These processes require energy. There are many mitochondria in the cell for a high rate of aerobic respiration. ATP is produced to provide energy. Mitochondria have a double membrane, with the inner membrane folding into cristae. The glycogen granules and lipid droplets are for energy storage. Glycogen forms from many glucose residues. They are hydrolyzed to release glucose whenever glucose is needed in the cell. Lipid droplets can provide fatty acid. They also provide components for cholesterol synthesis. 2 wants you to state one difference visible in figure 3.2 between a peroxisome and a mitochondrion. Mitochondria in the diagram have a double membrane. You can see the foldings of the inner membrane in the photomicrograph. Those foldings are called cristae. Peroxisome does not have it. The peroxisome is spherical, and mitochondria show a variety of shapes. You can also describe the shape of a mitochondrion as a sausage shape. 3. Suggest why a mitochondrion can synthesize enzymes, but a peroxisome cannot synthesize enzymes. One special feature of mitochondria is that they contain circular DNA and 70S ribosomes. The circular DNA contains genes that code for enzymes needed in the organelle. 70S ribosomes can be the site of translation. It also has the enzymes needed for transcription and translation to take place. This leads to the synthesis of enzymes. Examples of such enzymes include RNA polymerase and tRNA activating enzymes. Since it is the site of aerobic respiration, it can produce ATP needed for enzyme synthesis. They accept 2 marks for AVP. You can get full marks if all your points are made up of the AVPs. For example, mitochondria have proteins called chaperones. They are involved in the folding of polypeptides to give the tertiary structure. Besides, ATP cannot enter peroxisome to be an energy source. Energy requiring processes such as protein synthesis cannot take place. 4 wants you to suggest why it is useful to the cell that the breakdown of hydrogen peroxide occurs within peroxisomes. Hydrogen peroxide can be toxic to cells. Since peroxisomes are membrane-bound, reacting inside can protect the rest of the cell from hydrogen peroxide. The process of separating the interior of a cell into distinct compartments, each with specific conditions, is called compartmentalization. Peroxisome act as a compartment specifically for this process, so it is more efficient and controlled. This is because a high concentration of catalase is contained in one location. Besides, the conditions required for the enzymes to work effectively can be controlled inside peroxisomes. Optimum conditions, such as suitable pH, can be provided for the reaction. Oxygen is produced as a byproduct of H2O2 decomposition. It is useful for peroxisome reactions that require oxygen. It is mentioned in Part C that some processes occurring in peroxisomes need oxygen. There is a mark for AVP. For example, this compartmentalization can isolate peroxisome enzymes from the rest of the cell. 4a. Explain how blood flow through the alveolar capillaries helps to maintain steep diffusion gradients for gas exchange. A steep diffusion gradient is a large difference in concentration between 2 areas. In this case, we want a constant higher oxygen concentration in the alveolar space and a higher concentration of carbon dioxide in the blood. The blood arriving at the alveolar capillaries is deoxygenated. It comes from the right ventricles, so it has a lower partial pressure of oxygen and a higher partial pressure of carbon dioxide. This allows oxygen diffuses into the blood and carbon dioxide in the opposite direction. The newly oxygenated blood is constantly removed from the area. The oxygenated blood is passed to the pulmonary vein, so the oxygen that diffuses from the alveoli into the blood is transported away continuously. This prevents equilibrium from occurring. B. Explain the role of elastic fibers in the alveolar wall during ventilation. Elastic fibers show great elasticity and resilience. When air enters the alveoli during inhalation, it allows the alveoli to expand or stretch. This prevents alveoli from overstretching or rupturing due to the increased air volume and pressure. During exhalation, they recoil to force the air out. Note that you cannot replace the term stretch and recoil using contract and relax. Those words are used to describe the action of smooth muscles, not elastic fibers. C talks about an organelle called the lamellar body that stores surfactant. ABCA3 is a protein needed to move surfactant phospholipids into lamellar bodies. You are asked to suggest and explain the features of ABCA3 that make it suited to its function. To transport substances into lamellar bodies, we need to move them across the membrane surrounding the organelles. Even though this is not about the cell surface membrane, it is very similar to how substances move through it. With that said, ABCA3 acts as a transport protein. Since it transports phospholipids, which are large, it is a carrier protein rather than a channel protein. A carrier protein has a binding site specific to the substance it transports. The binding leads to a conformational change of the carrier protein. To store a reasonable amount of phospholipids in the membrane enclosed structure, it is not likely that we can just depend on the passive movement of the substance. Active transport is needed, and ATP is used. Surfactant phospholipids move against their concentration gradient. To store a large quantity, we do not want the process to stop at equilibrium. D1 shows the sequence of events for the production of ABCA3. The sequence of nucleotides in the gene is used to produce the primary transcript in a process called transcription. The sequence of nucleotides in the mRNA will then be used in translation to produce the polypeptide. D2 wants you to suggest why the number of base pairs in a gene is way more than the amino acid it codes for, apart from the fact that a triplet base codes for one amino acid. They are introns and exons in our DNA. Introns are the non-coding sequences that do not code for amino acids. They are not involved in polypeptide synthesis. They are removed from the primary transcript to form the mature mRNA during the post-transcriptional modification. Several events occur during the modification. The one described here is called RNA splicing. After the introns are removed, exons are joined to form the mRNA. Apart from that, some DNA triplets are transcribed into the stop codon in mRNA. The stop codons are UAA, UAG, and UGA. They do not code for amino acids. Methionine, the first amino acid coded for by the start codon. It is usually removed after translation. The start codon is the first codon in mRNA and is usually AUG. There is a mark for AVP. You can talk about the other non-coding regulatory sequences in DNA which do not code for amino acids. For example, the promoter sequence serves as the binding site for RNA polymerase. Question 5 is about TB. Mycobacterium bovis is the species of bacterium that causes the transmission of TB from dairy cows to humans. Remember to capitalize the genus name. B. Outline a control measure that can be taken to protect people who consume milk and dairy products from these infected cattle. Mycobacterium is sensitive to heat and can be killed by heat that is at least 65 degrees Celsius for at least 30 minutes. To ensure the safety of milk, a process called pasteurization can be carried out. It is a process by which milk is heated to a specific temperature for a set period to kill pathogenic bacteria. Raw milk or unpasteurized milk should be boiled before drinking. Pasteurized milk should be used to make dairy products. Meat from dairy cattle should be cooked thoroughly to ensure mycobacterium are killed. C. Explain the most likely mode of transmission of TB from an infected person to an animal. Your answer to this question should contain two parts: how the infected host releases the pathogen and how it enters the uninfected host. The method is very similar to how it is transmitted from human to human. The infected person coughs, breathes out, talks or sneezes and releases aerosol containing the pathogen. The uninfected animal inhales aerosol or droplets that contain the bacteria. D. Outline the treatment that is used to kill Mycobacterium tuberculosis in latent TB infections. It can be treated by antibiotics, as it is caused by a bacterium. A combination of antibiotics is usually used. This can remove the pathogen more effectively and prevent the arising of antibiotic resistance. This is because some Mycobacterium have shown resistance to some commonly used antibiotics. Using a combination of antibiotics prevents any survivors which may start developing antibiotic resistance. Providing any named antibiotic can give you the next mark. The treatment usually continues for 4 to 12 months. This is because Mycobacterium is a slow-growing bacterium, making the treatment less effective. Using a combination can shorten the time to complete the course of antibiotics. A single antibiotic treatment that lasts longer increases the chance that an individual stops the treatment halfway. This can contribute to the development of antibiotic resistance. Vaccination cannot be accepted as an answer because it is not a treatment when one has already been infected. E. With reference to the structure of the wall of elastic arteries, suggests how damage caused by Mycobacterium tuberculosis infection can lead to the rupture of the artery. Elastic arteries have 3 layers: tunica intima, made of a single layer of flattened endothelial cells; tunica media, made of smooth muscle cells and connective tissue, with a large proportion of elastic fiber to smooth muscles compared to muscular arteries; tunica adventitia, the outer layer that contains collagen fibers and elastic tissue. The rupture of an artery is usually caused by damage to its tunica media and tunica adventitia, which are the main supportive layers. It is possible that the bacterium causes damage to the elastic fibers in tunica media or collagen fibers in tunica adventitia. Without the elastic fibers, the artery loses its elasticity and the ability to stretch. The artery will lose its ability to withstand the increase in blood pressure. The wall may be overstretched or overexpanded, leading to bursting. It also cannot recoil properly as there are fewer elastic fibers. Figure 6.1 shows the requirements for the efficient transport of water from the roots to the leaves. The specialized cells arranged end-to-end to form xylem vessels are called xylem vessel elements. The question wants you to name the cell, so the tissue name xylem alone will not be credited. B. Explain how tension is created in the xylem vessels. The upward movement of water is due to the transpiration pool. Water evaporates from the cell wall of spongy mesophyll during transpiration. This leads to a low water potential near the leaves. A water potential gradient is created. There is a low pressure at the top of the plants near the leaves and a high pressure at the bottom near the roots. You can also say that it is the hydrostatic pressure difference. Cohesion and adhesion of water molecules allows water to move up as a column. Cohesion is the attraction between water molecules. It occurs because of the dipole property of water. Adhesion resulted from the hydrogen bonding between water molecules and the cellulose cell wall. It can also occur with the hydrophilic parts of lignin in the cell wall of xylem vessels. C1 wants you to explain how the lignified wall is important for the efficient transport of water. Due to the upward movement of water, there is pressure exerted from the outside. Lignin prevents inward collapse or compression from the tensions created. Lignin is rigid and strong. It provides mechanical support to the cell wall. It forms a secondary thickening for this purpose. Besides, the adhesion of water to the hydrophilic groups in lignin occurs to maintain the column of water. Note that lignin is generally hydrophobic. So, it is important for you to highlight that adhesion only involves its hydrophilic part. As a generally hydrophobic substance, lignin has waterproofing properties. This prevents water from leaving xylem vessels. They can only move out at the unlignified parts called pits. Lastly, lignification is associated with cell death. Vessel elements lose their cytoplasm and organelles to form a hollow tube. There is a mark for AVP. For example, lignin causes the cell wall to be rigid. Without the ability to stretch and expand, tension can develop inside. 2. Describe one other main change that needs to occur to these cells so that their structure becomes suited to their function. Xylem vessel elements die when they mature. It occurs in a process called apoptosis. Apoptosis is also known as programmed cell death. It is a natural process in which cells die in a controlled manner. When they die, they lose cell contents, including cytoplasm, nucleus, and other organelles. There is a mark for AVP. You can talk about the formation of pits, which are the unlignified part of the cell wall. It allows lateral movement of water to the surrounding cells. That's all for today. If you think my videos are useful, don't forget to like, comment, and subscribe. Thank you for watching and see you again soon.